right, we're now in section 4 of chapter 16 on the senses with visual receptors. And learning objectives for this section are to describe the accessory structures of the eye and list their functions, describe the structures of the eye, describe refraction of light, discuss how light is focused on the retina, define phototransduction, and to compare and contrast the two general types of photoreceptors, including their photopigments. Not all of which we're going to actually be covering. Ideally, in covering the whole section through the text, you'd be going through a lot of this information. We're not going to cover all of it because it's a rather substantial section. As well, explain the bleaching reaction, which we won't technically cover, but ultimately, long term, you should know how it relates to dark adaptation, light adaptation, and describing the visual pathway from the photoreceptors to the brain, and explain how stereoscopic vision provides depth perception. And I went past the eyebrows and the eyelids for some of the basic information that I think you can know without necessarily have to go through it in great detail. But starting with the conjunctiva, which is the transparent lining of the eye and lid surfaces. It includes a specialized stratified columnar epithelium. And it doesn't cover the cornea, so as not to interfere with light passage, and pink eye is really referred to as conjunctivitis because it affects the conjunctiva. And of course, you should know, generally speaking, that contains numerous goblet cells to moisten the eye and blood vessels to nourish the sclera and abundant nerve endings. You should definitely know that. That might just show up on your exam. This is on your exam, the external anatomy of the eye and surrounding accessory structures in the sagittal section. Certainly noting that the different areas here, the levator palpebrae here, palpebrae, or eyebrow, of course, orbicularis oculi muscle. Get a little closer. You're looking at right towards the sagittal plane. Look right here. Conjunctival fornix here, and the conjunctival fornix, and the ocular conjunctiva, the palpebral conjunctiva, the tarsal plate, the eyelashes, our palpebral fissure, where we have, of course, our pupil here, the lens, our iris out here, our cornea, and our sclera, okay? And eyelid, tarsal glands, orbital fat, and orbicularis oculi muscle, okay? You definitely will need to know that. Generally speaking, you know the lacrimal apparatus produces, collects, and drains fluid. That might show up in your exam. It lubricates, cleanses, and moistens the eye, and reduces eyelid friction, defends against microbes, and oxygenates and nourishes the cornea. And that's not specific for us right now. Lacrimal apparatus, this diagram will be on your exam. Lacrimal gland being here, the orbital part. Lacrimal gland, palpebral part. Lacrimal puncta here. And then we have our lacrimal caruncle, our lacrimal caniculi, our lacrimal sac, our nasal lacrimal duct, our nasal cavity, and our nostril. This will also be on the exam. Tunics of the eye, the fibrous tunic, the sclera, the cornea, and this is repeating in numerous images. You may see these quite a bit, so I want to make sure you know them. The iris, the ciliary body, the choroid for the vascular tunic, and the retina with the pigmented layer and the neural layer. As well, you'll have a diagram of the internal eye structures, noting the optic nerve sheath, and the CN2 optic nerve through here, the central artery of the retina, and the central vein of the retina, optic disc, our fovea centralis, which is a big component. That's important. We'll talk more about that with more content for your exam as well in a little bit here. The retina, our choroid. If they're taking away layers here, right? The sclera. Here we have the posterior cavity, which contains the vitreous humor, all the way through here. Our posterior chamber, our anterior chamber, this gray area through here, including our anterior cavity, which contains both the posterior chamber and the anterior chamber. And this contains the aqueous humor. The sphincter pupillae, or pupillae, and dilator pupillae. Our pupil, once again here, and our cornea here. Our iris, right, our lens, our scleral venous sinus, limbus, our suspensory ligaments, our ciliary process, our ciliary muscle, part of the ciliary body, and our ora serrata here. So you certainly should generally know, of course, the fibrous tunic is the tough outer layer, the sclera is the white of the eye, the cornea is anterior convex transparent window, where it refracts the light. Ultimately have to know the location of these areas, but not going to specifically have to know their functions for the exam of the vascular tunic areas. Control of pupil diameter. You certainly should know the pupillary construction. Bright light, the sphincter pupillae is going to contract a parasympathetic innervation, and so it dilates. I'm sorry, it constricts. The pupillae dilation, low light, we're actually going to see the pupillae contracting and sympathetic innervation and dilates. Okay? And the retina, of course, is the neural tunic and contains the pigmented layer, the neural layer, and the ora serrata jagged edge. So you just have to know the locations of these right now. The retina, the cells of the layer form 3 sublayers, the photoreceptor cell, we have our bipolar layer, and our ganglion cell. You're going to have to know more about the rods and the cones shortly. We're going to talk about that. This will be on your exam, the structured organization of the retina. Noting here the optic nerve, our retina, our sclera, our choroid, that red and pinkish layer there, our optic disc here, our fovea centralis, okay? And again, in a bigger image here being the choroid, the rods, and the cones, our horizontal cell, our bipolar cells, our amacrine cell, our ganglion cells, axons of the ganglion cells, the optic nerve, and the part of the posterior cavity, and our pigmented layer here for the retina, and the neural layer of the retina. Okay, know the difference between, you don't have to know the difference between ECME light and nerve signal response, but that's showing you, of course, to be able to see that coming both ways. The components of the retina, again, just have to know the location for the optic disc and the macula lutea, certainly not as much of the fovea centralis component of there, which is certainly a big component for us, both in terms of your location as well as an area we're going to talk about here with the rods and the cones. As you know, the optic disc is the blind spot. Just generally know that. The lens, of course, changes shape to focus light on the retina. That's part of a question on your exam, talking about the rods and the cones, which we'll talk about, as well as accommodation. When you're viewing objects closer to 20 feet, we're looking at accommodation, where the muscle tenses, suspensory ligaments are less tense, and the lens is more spherical. That is on the exam. You need to know that. And this diagram will be on the exam. The ciliary muscles, the lens flattened here, lens shape for distant vision. Suspensory ligaments are taut here, okay? Whereas lens shape for near vision, or the accommodation, the ciliary muscles are contracting, moving the cellular body close to the lens. The lens is thickened with more spherical here, and the suspensory ligaments are relaxed. As far as the structure, again, noting more the location of vitreous humor and aqueous humor. Generally speaking, that vitreous humor, of course, helps maintain the eye shape. It supports the retina, keeps it flush against the back of the eye. The aqueous humor nourishes and oxygenates the lens and the inner cornea. I'm not worry about that more detail right now. That image, just trying to recall, that image was not on your exam. The refraction of light, certainly know that sharp vision requires light rays to be bent and refracted as they pass toward the retina. Refraction results when light passes. Now, we start talking about rods and the cones here in a moment. We're not going to worry too much about the combination of the lens. Certainly, you know the cellular muscle contraction thickens the lens, with the slackened suspensory ligaments allowing the lens to thicken, and refraction increases, as we mentioned. Okay. Whereas convergence of the eyes, the extrinsic muscles pull the eyes medially and directs the image of interest into both foveas, and the weak eye muscle may cause diplopia, or double vision. Not going to worry about phototransduction specifically. Photoreceptors, the rods and the cones. This information you need to know, all of this. The rods are longer and narrower than the cones, are more numerous. Each rod is highly sensitive, activated by the dim light. The periphery of the retina contains many rods. Those rods converge on fewer bipolar cells, which converge on fewer ganglion cells, and results in sensitivity to dim light, but a blurry image. And the cones are concentrated at the fovea centralis, activated by high-intensity light, and they allow color vision. Expect to have to be placing these words into sentences with putting the right words in the right places. It's the kind of exam question you're going to get with the rods and the cones. And it's kind of cool, noting here the same detail that you need to know, less the image. The image is not going to be there, but certainly this content right here you should know. The rods being more numerous, primarily located in the peripheral retina, specialized for dim light, night vision, can't distinguish color, poor sharpness of vision. The cones are less numerous, located in the fovea centralis. They respond to stimulation by bright light for color recognition and sharpness of vision, and they're subdivided into blue, green, and red cones to see different frequencies. Not going to actually worry about this specifically for the exam with photopigments. Dark and light adaptation you should know. The content will kind of be related to the content of the exam, how the return of sensitivity to low light levels after bright light, the bleached rods have to regenerate rhodopsin, which is basically in the cones or the rods. It may take 20 to 30 minutes to see well. Light adaptation, the process of adjusting from low light to bright conditions. Pupils constrict, but cones initially are overstimulated. It takes about 5–10 minutes for full adjustment. In the nerve signals, just know in the dark, rods are depolarized, and basically glutamate hyperpolarizes the bipolar cells, preventing them from exciting the ganglion cells. This image is on your exam, noting for our right versus our left eye here and binocular vision between there. We have our uncrossed ipsilateral axon, our crossed contralateral axon, projecting fibers of optic radiation here. Our retina, of course, here, our optic nerve, optic chiasm, optic tract. Here's our lateral geniculate nucleus of the thalamus, our pretectal nucleus of the midbrain, our superior colliculus of the midbrain, and our primary visual cortex, the occipital lobe. Okay, that'll be on the exam. The eye is very extensive. We only covered a very small amount of it, trying to keep it specific for you because it's just too much information right now. If you're going to be an optometrist, an ophthalmologist, or some area of optics, you'll want to know that in much greater detail. That being said, what'd you learn? Where's the conjunctiva located and what are its functions? How is lacrimal fluid spread across the eye surface and removed from the orbital region? What are the 3 eye tunics and what's the primary function of each tunic? And compare the anatomic structure of the cornea and the lens. Explain how oxygen and nutrients are supplied to these 2 structures. What are the functions of vitreous humor and aqueous humor? Describe how lights focus on the retina when viewing an object closer to 20 feet. What are the differences between the rods and the cones, which is definitely on the exam, as well as accommodation with less than 20 feet. Respect their anatomy, their photopigments, and how light they process. How does dark adaptation differ from light adaptation? What occurs during phototransduction of light? Which area of the brain consciously perceive visual stimuli and which areas respond reflexively below the conscious level? And what's the significance of some ganglionic axons crossing the opposite side of the brain? Our last section is hearing and equilibrium, and we'll come back to that shortly.